In this video, we will discuss about one of the sensor that can detect this mesenchymal to epithelial characteristic or gene expression, and also it can detect this epithelial to mesenchymal gene expression. Not actually the gene expression. This sensor can help us to detect the change in the glycomics profile when there is a change in the phenotype of the cells, that is from mesenchyms to epithelials or from epithelial to mesenchyms. So we will understand this sensor, and in parallel we will understand uh, how this ZEB1 protein helps in this mesenchyms to epithelial and epithelial to mesenchyms transfer. So the normal function of this ZEB is to suppress the epithelial genes. So it will promote or therefore it will promote the cells to behave like a mesenchyms type, as it will suppress the genes that helps in the epithelial phenotype of the cells. And in the process of this ZEP protein formation, there is a post conformational changes occur in the 3' un, uh, untranslated region of the ZEB, and this untranslated region of the ZEB will get affected by small microRNAs, and one of the microRNAs that are used in this plasmid or in this arrangement is this microRNA 200. So this microRNA 200 will disrupt this untranslated region of this ZEB protein, thereby makes this ZEB protein unstable. And when the ZEB protein is unstable, there is formation. This leads to the inhibition of the ZEB protein formation. Thus, the suppression of epithelials genes will get disrupted, and cells will start to behave more like epithelial cells. So, first, how this sensor will work? First, we will transduce this sensor so that it get integrated with the host genome, and once it get integrated with the host genome, we will transfect this plasmid, and this plasmid is doxycycline derived for this microRNA 200. So when this plasmid is transfected into the cells, and when doxycycline is introduced, then this microRNA will starts to form. When this microRNA will starts to form, it will disrupt this 3' 3' untranslated region of the ZEB, and it will disrupt the actual ZEB proteins mRNA, and also it will disrupt this plasmid translation. So this ZEB3 untranslated region plasmid translation will get disrupted, thereby this it will not leads to the translation of this this destabilized DS-RED, and also this SYN4H translation will also not occurs because of the unstabilization of this 3' ZEB 3' untranslated region because of this microRNA 200C. So it will work at two sites, for a general understanding, that one site is for that it will disrupt this sensor, and also it will disrupt the actual ZEB1 protein. And when the ZEB1 protein will get disrupted, it will the suppression of the epithelial genes will not occur, and this E-catechorin is one of the proteins for the epithelial genes, so it will starts to translate it. And when this E-catechorin will starts to translate it, this SYN4H will also translate it along with this green fluorescence protein, destabilized green fluorescence protein, so that we will start to see the green signal that indicates that the cells are more close to the epithelial like characteristics. And also this SYN4H will also get translated from this side, and this SYN4H is the we can say is the reporter for the intracellular glycomics profiling, as there are 3 to 4 N glycan sites and also the O glycan sites that can be that can be used to identify whether there is a change in the glycomics profiling occurs or not by analyzing the Syn4H glycans or glycans that are attached to the Syn4H glycan sites. So this is how this EMT, these sensors can help us to or identify the change in glycomics profile when the cells has this epithelial-like characteristics. However, let's consider the other case when the doxycycline is not introduced, then ZEP1 will continue to inhibit epithelial-like cells, epithelial genes. So this E-cadherin will not be translated as it is epithelial genes, and ZEP1 will suppress the translation of this E-cadherin genes. So the cells will not show green color, or green color will starts to fade, and after some time when this ZEP1 will get stabilized, then this region will starts to translate it of the sensors, and in this region this Syn4H will be translated with the help of the because of this CMV promoter, and simultaneously the this destabilized DS red will also get translated. So now the cells will show the red color, red fluorescent signals. It is the characteristics of this mesenchymal phenotype of the cells, and in this case the Syn4H will help us to understand, or will help us, or will provide the insights about the change in the glycomics profile when the cells has this mesenchymal type phenotype. So this is how this sensor can be used, not just to visualize whether the cells have more epithelial or mesenchymal like characteristic, but this it also contains the glycomics reporter that help us to know whether the change in the glycomics profile within the cells because of the change in this epithelial to mesenchymal or mesenchymal to epithelial phenotype. So I hope you got some idea how this sensor help us to understand and provide more insights about the change in the glycomics profiling within the cells of different phenotype. If you need to understand more about this epithelial to mesenchymal or mesenchymal to epithelial type sensor, you can either go through the reference or provide me any specific questions in the comments section, as so far this sensors this sensors research is not yet published, or I'm not aware whether any study has been published based on this sensor. That's all for this video. Thank you for your time.